Before telling you about sort of what I imagine as some pathways toward developing novel therapeutics in autism, I think it's important to note that in order to do that work, you sometimes have to work with companies. And I'll talk about a little bit of work funded by Novartis and Roche. And so that's potentially a conflict of interest. Maybe this is my attempt to be provocative, but there's a quote from Isabel Rappin, who shares this every year with the group of students at the Cold Spring Harbor course on autism. She says very clearly, autism is not a disease. And it's a little funny to think about that because we write this down on sheets of paper all the time as a diagnosis. It's certainly not something that you'd think about as a single disease. When I see somebody with autism spectrum disorder, I certainly don't think of them just as having that diagnosis. This conveys some part of that. So for an individual who's presenting to clinic, in the center you see the diagnostic criteria, but around the edges, so on the top, it may be more relevant that they have intellectual disability or language difficulty, at least at the point in time when I'm seeing them. They may have a co-occurring mental illness, they may have a co-occurring medical problem that really is the focus of treatment immediately. Increasingly, people are either walking into clinic with a genetic diagnosis, and these are diseases. These can be defined much more clearly, not just on the basis of behavior, or they walk out with a genetic diagnosis, and we're just at the cusp of having some sense of what to do with those. On the bottom right, you see what we hope will become unifying factors, biomarkers, that will offer something in more than less than 1% at a time, which is really what's been found in the genetic domain. So when I see somebody with autism, they may fit this pattern, they may fit this pattern. If we have a study in autism, that may include 100 people, if we're lucky, 1,000 people, all fitting different patterns and probably representing many different diseases, but with common features in the center. So when we think about genetics, there's still a lot of data fitting this model, where you have multiple common risk factors. They frankly could be relatively rare risk factors that converge in an individual to lead to their risk of autism. In contrast to that sort of unifying model, most of the genetic literature thus far has favored something that feels more splintered, where you have individual genetic disorders that by themselves convey most of the risk for the individual. So Fragile X syndrome is one of the more common of these, maybe 1% of people with autism. Not everyone with Fragile X has autism, and that's important to note. But these are emerging over time where there are a few of them that are as common as 1%, but more of them that are considerably less common. Sometimes just a few people in thousands who have been assessed. As we accumulate more knowledge, Dr. Fishback mentioned 71 as our current number. That keeps growing, so day by day it feels like there are new autism genes, which is incredibly exciting. What we can hope is that those converge in some way, and there are some examples of that. I'm not going to spend a lot of time on that today, but those convergences could lead to potential treatments. So I feel like quoting Vizzini from The Princess Bride, we're just getting started at the level of complexity. One of the other challenges is that when a deletion or a disruption of a gene is implicated in autism spectrum disorder, frequently the duplication or excess function of the same gene or gene region is also implicated in neurodevelopmental disorders. And this is now so routine that people start looking for the duplication upon first identifying a deletion. And you can imagine the challenge there, that if you move one individual closer to health, you may take somebody on the other end of that inverted curve and push them into a state that's actually more difficult to deal with. So Dr. Fishbach really outlined the increasing complexity here on this inverted pyramid, where genes are relatively simple in comparison to the number of neurons. Now we're talking billions versus synapses where you're talking trillions or quadrillions. I had to look that up to make sure that's really the right term. You would hope, though, that as we get nearer the top of this pyramid, because we define autism at the level of behavior, that you should see convergence. So that maybe you're identifying brain regions or circuits that underlie social difficulties, say. And it probably isn't going to be as simple as, okay, well, this brain region is something that we know needs to be targeted in everybody with autism spectrum disorder, but that convergence is certainly what we hope for. What's emerged from the data to date is really convergence at the level of protein systems, convergence at the level of time points in development, with a substantial black box in between that and the behaviors that we use to define autism spectrum disorder. That is a substantial improvement on where we've been, okay? So this is the model that we've used for every treatment, basically, that is applied in autism spectrum disorder now, which is that we really don't understand how our treatments relate to the pathophysiology, the core biology in autism spectrum disorder. Instead, we're treating things as a black box. This is the example that I frequently use because this is the furthest we've gotten up the cascade. And I don't have time to go through all the details, but the example of Fragile X syndrome is really a beautiful example from a neurobiology perspective. This is work that was done at Roche in collaboration with Mark Baer's lab at MIT, where they were able to show that a drug that tunes down response at a particular glutamate receptor, when given shortly after weaning in a mouse, extending all the way to adulthood, that's 4 weeks in a mouse, results in a complete resolution of essentially everything that they can detect that's different in that animal. At the level of the brain, it results in a decrease in dendritic spines, which is the key thing that can be translated across species in Fragile X syndrome. Dramatic, amazing. Unfortunately, the behavioral parallels, and you're going to hear next from Dr. Crawley about behavior and how you assess that in models of autism, are really difficult across species, and particularly for the Fragile X model, that there isn't something you can say in the mouse is necessarily going to translate to the human. On the flip side, there were no obvious ill effects on health for using this drug, and this then can be translated at least to an initial test in autism spectrum disorder. You'd note there is still a black box. You'd also note not everybody with Fragile X syndrome has autism. So thinking about autism symptoms as the outcome may be a little fraught here. But we are quite far up that pyramid. And the question is, what happens when you translate to humans? And so this is not the punchline I'd like to deliver. There have now been failed trials from 2 different compounds, 2 different companies, in adults and adolescents, resulting in both Novartis and Roche closing their research programs. Some of you are scratching your heads and saying, well, is that really what we wanted to do? Did we want to study this in adults as our first swing and then move to adolescents? Or if we're studying a treatment that has potential in a neurodevelopmental disorder, do we really want to be thinking about children? And then the other concern is what outcomes were looked at. And what they did is they lumped what are termed aberrant behaviors together in a common outcome. Now, this is not an unreasonable thing to do. It was actually smart because the Food and Drug Administration said that's one of the things that they will potentially accept in a neurodevelopmental disorder. But really, when you're talking about dendritic spines, you want to be thinking about something that impacts on learning and studying learning in some way, likely earlier in development. So that's one example. The counterexample, and you could think of a number here, is targeting oxytocin. And so here, this is really thinking about the top on down, thinking about social behavior, things that move social behavior across species, and oxytocin clearly does that. Again, not the time to go into all the detail, but there is a little bit of emerging data in autism spectrum disorder where a few models either show changes in social behavior in response to being administered oxytocin, or in one model that Dan Geschwin's lab has worked on, you actually see abnormalities in the oxytocin system that can be rescued by administration of oxytocin. So there may be some connection down below, but really the thought here started from the behavior, thinking about core circuitry across species, not specific knowledge of the biology of autism itself. So lots of studies have looked at the acute effects of intranasal oxytocin in humans. A few of those studies have been in people with autism spectrum disorder, but there isn't data to suggest that this is specific to autism. If you give intranasal oxytocin to a person with autism, they're more likely to prefer to throw the ball to somebody who throws the ball back to them in a laboratory setting, okay? Cyberball task. That's not a treatment for autism, but it shows that this can change behavior in a laboratory setting with a single administration. And then this is data from Kevin Pelfrey's group at Yale showing that you also can see changes in brain region activation in response to social stimuli following administration of oxytocin in contrast to placebo. Those changes include brain regions that are important for reward, They also include some of the regions that Dr. Fischbach pointed out as belonging to what we think about as a social network in the brain. Again, very exciting. They didn't actually in this study see a change in behavior, however. So in some studies you can show that oxytocin, when administered through the nose, will change brain region activation, other studies behavior. Unfortunately, because this is something that to some degree is available, there are likely tens of thousands of kids in this country currently taking something that's at least marketed as oxytocin. A lot of this probably doesn't contain oxytocin. I put this up here a little bit in a lighthearted way, but this is not lighthearted, right? We need to know if this is something that could offer potential benefit. We need to study this in the real world. It might do something acutely in the laboratory, but you worry about administering something that we know acts on social brain systems, but not in a way that we understand terribly well in the human. And so we're part of a large network, and there are a number of studies out there to try to evaluate whether there are potential risks associated with treatment or whether some individuals might benefit. And it's important to say some. We don't expect that any single treatment is going to probably benefit the majority of individuals with autism when that treatment is anchored in biology because of the diversity. We talk about neurodiversity as defining autism spectrum traits apart from people without autism spectrum traits, but we also need to think about the incredible diversity within autism spectrum disorder itself. So it's important when we are thinking about translating treatments across species to think about who, and really you want to think there about where we are modeling, who we're modeling, and translate to individuals, say, who have Fragile X syndrome, instead of translating directly to autism writ large. We want to think about what, and particularly outcome measures here, and we need to think about when. So this is a neurodevelopmental disorder. We need to think about development when we're thinking about potential treatments. Where's the potential payoff? And I would say, having spent a lot of time with families of people with Fragile X syndrome and with people with Fragile X syndrome themselves, I would be thrilled to have a treatment that only worked in Fragile X syndrome. That would be great. But we also can learn from these studies how you study potential treatments based on an understanding of biology. We don't know how to do that in neurodevelopmental disorders. It's not been done. We need to figure out how to do that, and I hope that this work will allow some of that to happen. And then there's the question about whether some of this will extend. And I think that that is a reasonable hope, that if you find something that benefits 1% for sure, maybe there's another 5%, 10%, we can hope even higher than that, that may have common pathways implicated that may benefit from treatments. And that I think is the forward-looking hope. But again, being able to help 1% of the people who are walking into our clinics, 1% at a time, would be a tremendous advance. Thank you very much.